In a remarkable breakthrough that has captured the attention of the global scientific community, a team of researchers from the University of Warwick in the United Kingdom and Monash University in Australia has unveiled a powerful new antibiotic hidden within a bacterium that scientists have been studying for nearly 7 decades. This astonishing discovery marks a turning point in the ongoing fight against antibiotic-resistant bacteria, a challenge that has become one of the most pressing medical threats of the 21st century. The bacterium in question, Streptomyces coelicolor, has long been known to produce a wide range of antibiotic compounds that have shaped the foundation of modern medicine. However, despite years of exploration and research, the discovery of a completely new antibiotic molecule from this familiar microorganism has stunned experts and reignited hope for the development of next-generation treatments. The newly identified molecule, named methylenomycin C lactone, represents a hidden gem that was overlooked by scientists for decades. This discovery highlights how even the most well-studied organisms can still hold secrets waiting to be uncovered through advanced genetic and biochemical analysis. The research team achieved this breakthrough by carefully deleting specific genes within the Streptomyces coelicolor genome and observing how these genetic changes affected the bacterium's ability to produce antibiotic compounds. By mapping the intricate steps of its biosynthetic pathway, the scientists were able to isolate intermediate molecular components that had never been characterized before. Among these newly identified molecules, one stood out due to its extraordinary potency, being found to be 100 times more effective than methylenomycin A, the previously known antibiotic produced by this bacterium. This finding not only demonstrates the vast potential still hidden in microbial genomes, but also opens an entirely new chapter in antibiotic discovery. For decades, researchers have been warning that the world is running out of effective antibiotics, as bacteria continuously evolve resistance to existing drugs. This has resulted in a global health crisis where once treatable infections are becoming increasingly difficult to cure. The emergence of multi-drug-resistant bacteria, often referred to as superbugs, poses a severe threat to global health systems, leading to prolonged illnesses, higher medical costs, and increasing mortality rates. The discovery of methylenomycin C lactone could play a critical role in reversing this trend by offering a new weapon against these resistant strains. The significance of this discovery extends far beyond a single molecule. It underscores the power of genetic engineering and modern biosynthetic analysis in unlocking the hidden potential of microorganisms that have been part of human research for generations. Streptomyces species, which are soil-dwelling bacteria, are known to be prolific producers of natural antibiotics. In fact, more than 2/3 of the antibiotics used in medicine today have been derived from this group of bacteria. However, traditional methods of studying these microbes often focused only on the most easily detectable compounds, leaving behind a treasure trove of hidden biochemical pathways that could produce other, more potent substances. Advances in genomic sequencing and molecular biology have now made it possible to activate these silent or cryptic pathways, revealing compounds that were once thought to be inaccessible. This new antibiotic discovery is a prime example of how cutting-edge techniques can breathe new life into old scientific models. By deleting or manipulating certain genes, scientists can unlock pathways that have been dormant for decades, leading to the creation or identification of molecules with entirely new structures and properties. What makes methylenomycin C lactone particularly exciting is its structure and potential mechanism of action. Preliminary studies suggest that it may attack bacterial cells in a completely different way than existing antibiotics, meaning that pathogens that have developed resistance to current treatments may still be vulnerable to this new compound. This is exactly the type of breakthrough the world needs to overcome the growing antibiotic resistance crisis. Antibiotic resistance occurs when bacteria adapt to survive exposure to drugs that are designed to kill them. Overuse and misuse of antibiotics in both medicine and agriculture have accelerated this process, turning once powerful drugs into ineffective treatments. The discovery of a molecule that is not only new, but also significantly more potent, represents a critical opportunity to develop next-generation therapies that could save millions of lives. Scientists at Warwick and Monash believe that their work demonstrates a new way forward in antibiotic discovery, one that involves revisiting well-known microbial species using advanced technologies and modern analytical tools. Instead of searching for new bacteria in unexplored environments, researchers can now re-examine familiar species with fresh eyes, uncovering hidden potential that has remained untapped for years. This strategy could rapidly expand the pool of potential antibiotic candidates and shorten the time required for development and testing. Beyond its immediate medical implications, this discovery also carries profound industrial and scientific significance. The pharmaceutical industry, which has struggled to keep up with the pace of antibiotic resistance, could benefit immensely from such innovations. Developing new antibiotics is notoriously difficult and expensive, with many companies abandoning antibiotic research due to low profit margins and high risks. However, discoveries like methylenomycin, lactone reignite interest in the field by proving that groundbreaking compounds are still waiting to be found, even within bacteria that have been studied since the mid-20th century. Moreover, the collaboration between the University of Warwick and Monash University highlights the importance of international cooperation in scientific research. Complex challenges like antibiotic resistance require a united global effort, combining expertise in genetics, chemistry, microbiology, and bioengineering. Through shared knowledge and resources, scientists can accelerate the pace of discovery and create solutions that benefit humanity as a whole. The process that led to the identification of methylenomycin lactone is a testament to this collaborative approach. By integrating advanced genetic editing techniques, high-resolution spectroscopy, and chemical synthesis, the team was able to pinpoint and characterize molecules that were previously invisible to standard methods. The discovery also serves as a reminder that scientific progress often involves patience, persistence, and a willingness to look deeper into familiar territory. Streptomyces coelicolor, despite being one of the most extensively studied microorganisms in microbiology, has proven that nature still holds countless surprises. For over 70 years, scientists believed they had mapped its antibiotic potential completely, but this latest finding shows how much remains to be discovered when we apply modern technology to long-established systems. Looking ahead, the next phase of research will focus on testing the clinical potential of methylenomycin lactone and its derivatives. Scientists will need to determine how the molecule behaves in living organisms, how safe it is for human use, and how it can be synthesized efficiently for large-scale production. If successful, this discovery could lead to the development of a new class of antibiotics that will redefine modern medicine. The fight against antibiotic resistance is far from over, but breakthroughs like this bring renewed optimism to the scientific world. In a time when many feared that the antibiotic pipeline had run dry, this finding proves that innovation and curiosity can still uncover life-saving solutions. The potential impact on global health is immense, from treating drug-resistant infections to preventing outbreaks of untreatable bacterial diseases. It also reinforces the need for responsible antibiotic use to ensure that new discoveries remain effective for future generations. In summary, the discovery of methylenomycin lactone within Streptomyces coelicolor stands as one of the most exciting scientific achievements in recent years. It blends decades of microbiological research with the precision of modern genetic science, showcasing how humanity can continue to outsmart nature's challenges through perseverance and ingenuity. This finding not only opens the door to new therapeutic possibilities, but also symbolizes hope, a reminder that even in a world facing a growing crisis of antibiotic resistance, the solutions we seek may already exist, waiting quietly within the microscopic life forms that surround us. This new antibiotic discovery has the potential to revolutionize medicine, protect millions of lives, and redefine our understanding of microbial potential. Antibiotics, bacteria, Streptomyces coelicolor, methylenomycin lactone, University of Warwick, Monash University, antibiotic resistance, medical breakthrough, scientific discovery, pharmaceutical innovation, genetics, microbiology, and biotechnology, antibiotics.